Hello, my name is HourtovenkoC. I'm a biology master's student at Laurentian University, and today I'll be presenting my research titled Effects of High-Dose Gestational Irradiation on Fetal Brain Development. I would like to state that I have no relations with any for-profit or non-for-profit organization, and this session has not received any financial or in-kind support. This study investigates how high-dose ionizing radiation exposure during pregnancy can affect fetal brain gene expression. Accidental exposures to radiation, such as those seen in nuclear disasters, such as the nuclear bombings of World War II and the Chernobyl nuclear reactor meltdown, have been shown to cause significant neurodevelopmental damage among children exposed. However, there is limited research on prenatal exposure and its effects on offspring's brain development. And there's even less research looking into the gene transcription across the different brain regions within the developing fetus. So to explore this, we used pregnant bulb C mice, divided them into two groups. First group is our sham group, which received no irradiation. This is our control group. And then the second group is an irradiated group, which received 1.8 grays of X-ray radiation on gestational day 14. On gestational day 18, the fetuses were then euthanized, measured, and their brains collected, then flash frozen. We then performed whole transcriptome RNA sequencing on the 4 dissected brain regions, the telencephalon, diencephalon, midbrain, and hindbrain. And then over 15,000 genes were then analyzed to identify our differentially expressed genes, or also known as our DAGs. Among the irradiated group, we saw a significant decrease in fetal size in regards to the weight, biparietal diameter, and crown rump length. Among the 4 brain regions, the telencephalon did show the highest number of DAGs, with an average of 1,882 dysregulated genes between male and females. And then the midbrain showed the next highest number of DAGs, with only about 75, indicating the difference in sensitivity within the telencephalon to radiation in comparison to the other brain regions. And then importantly, sex differences were evident in which genes were affected. Looking at the pathway analysis, we found that the most significantly dysregulated pathway within both male and females in the telencephalon was the complement and coagulation cascade. This suggests an immune and inflammatory response from the radiation. Other top pathways include the cytokine signaling and ECM receptor interactions, and that can all be found within figures 1 and 2. We also completed a gene ontology analysis to determine the dysregulation within biological processes, molecular functions, and cellular components, where the top 5 dysregulated for each can be found within figures 3 through 8, where there is actually some overlap between the sexes. To summarize, high-dose gestational radiation exposure alters gene transcription across multiple brain regions, with the telencephalon being the most affected, where both structural and functional pathways were impacted. These changes may contribute to long-term developmental dysfunction. Each region and sex responded differently, emphasizing the complexity of radiation's effects on the developing brain. We would like to thank Bruce Power, the Nuclear Innovation Institute, and NHRC for supporting this work. Thank you very much for listening.